right, I'll hang out up here.
Again, I want to thank our gold sponsors, Mass, Audubon, and Manomet, and our bronze sponsor, Avian Research Supplies. And I also want to thank Wildlife Acoustics, the Cornell Lab of Ornithology Birds of the World, and Feather Friendly. We welcome everyone to AFO100 and celebrate diversity in all its forms. AFO100 is dedicated to providing an inclusive environment that is collaborative, supportive, and engaging for everyone involved, and that is free of discrimination, harassment, and retaliation. Thus, we expect all meeting participants to adhere to the Code of Conduct throughout the conference. In the hopefully unlikely event that the code is breached, we ask that individuals report incidences immediately to the AFO SAFE Committee at safeafo100@googlebooks.com. Alternatively, incidences can be submitted anonymously to the comment box at the silent auction, along with any comments, concerns, or questions for the AFO Ethics Committee. Just a few highlights for today. We have a special event, the Wildlife Acoustics Workshop. If you want to attend, that was through a pre-register, so you had to pre-register on CVent, and it's from 1 to 2 in the Standish room downstairs. We also have meet the Journal of Field Ornithology editorial board from 2 to 3 in the Pimpton room. And we're moving the silent auction tables to here, the corridor actually. The final bids are going to go from 4:45 to 5. It says here Atlantic room, but it's not going to be in the Atlantic room, it's just going to be here. And Jen Smith is going to be sending emails out between 5 and 6 p.m. And then she will be at the silent auction from 5 to 7 p.m. And we accept only cash or check. And if you're not there to receive your item, the item will go to the next highest bid. We have a documentary special event, A Naturalist in the Rainforest, a portrait of Alexander Scotch at 5 p.m. in the Wampanoag room. And then we have also the banquet dinner and awards ceremony. And I also want to let all of those that might be interested in the educational workshop that's going to be happening tomorrow, that we are working on a location. There was a little bit of a misunderstanding of where it will happen, and we'll let you know through social media, and we might post that on at the registration desk also, where the location will be. So if you're planning on attending the workshop, just check the registration desk or check our social media platforms, and we'll inform that there. We have and I hope that it will still be there, we have a banner that it's up at the Atlantic Room, and please, it's your last opportunity to take your photo. You can take your photo, crop it, and then rotate it and post it on social media, and you can use the hashtag AFO100. We want to see who's part of AFO's flock, actually, and so please do that, and we can flood our social media platforms with your photos. And I wanted to uncover the new JFO cover, but we cannot find the foam board with the cover, so I'm just going to do this here. So we have a new cover for the Journal of Field Ornithology, and we actually had a competition going on where we asked our members to name the species on the JFO cover. And so if Megan, Annika, Miko, Jen, Gabi, or Lynn are in the room, please come see me after the plenary. We have a t-shirt for you. And if they're not, the t-shirts will go to the next ones, Kelly, Tori, Meredith, and Leah. So please come see me if you're on that list right after the plenary, and we'll make sure you'll get a t-shirt. And with that, I'm going to ask Brian Harrington to introduce our plenary speaker. Thank you.
Well, thank you, Valerie. It's a pleasure to be introducing Dr. Katharine Parsons. I've known Katharine since the late 1970s, when she was a student intern at what was then called Manomet Bird Observatory. She became an integral part in the study of a heronry on Clark's Island in Plymouth Bay, and soon accepted leadership of that project. While earning and after receiving her PhD at Rutgers University, Katharine returned to Manomet to lead her colonial waterbird studies of the East Coast, eventually becoming one of the first to document sublethal effects that organophosphates were having on colonial waterbirds. She has published more than 45 peer-reviewed scientific papers. So after leaving Manomet, Dr. Parsons became head of the Colonial Waterbird Program of the Massachusetts Audubon Society. She worked to grow partnerships with governmental agencies at local, state, and federal levels, leading to one of the most successful recovery programs for an endangered species, the piping plover. In the 1980s, there were just 135 nesting pairs in Massachusetts. Today, there are over 800, approximately 45% of the entire Atlantic coast population. The Coastal Waterbird Program has also successfully bolstered numbers of several kinds of terns, oyster catchers, and has been a strong participant in the recovery of other coastal birds, such as common and endangered roseate terns. So with that, it's a pleasure and a privilege to be introducing Dr. Katharine Parsons.
Well, Brian, thank you so much for that kind introduction. I guess is we allowed to take our masks off? Okay. It's a huge pleasure for me to be here with all of you today at the 100th anniversary of the Association of Field Ornithologists. I feel like I am one of you. As Brian said, I've been working with and studying birds in the field for a very long time, many decades. So this is just a fantastic opportunity for me, and I feel very honored to be here among you. I want to thank Valentina in particular for the kind invitation to be here and to share with all of you some of the work that I've conducted since the early 1970s. I'm just going to change this a little bit. There we go. All right. Okay. So just to start at the very beginning, in the early 1970s, I was invited, I was a high school student, to join a heron nestling banding expedition in Rhode Island. I was useful in climbing trees, which was kind of where I got my start. And so my interest in these birds has spanned from that time to the present, where I'm very happy to be conducting a study with colleagues at Mass Audubon on herons that are nesting in Boston Harbor. So the majority of what I'll share with you today is actually from studies that I conducted while I was a staff member at Manomet, but there is a little bit of information that's more recent. Here we go. And over the decades, as you can imagine, since it's been a long time, I had the opportunity to work with many generous colleagues and mentors. And I want to say in particular that Brian Harrington took me on as an intern way back in the 70s, and he showed me the importance of always asking questions and questioning everything, basically. It was great to sit in on staff meetings with Brian and have everything questioned. Excuse me. But I think more importantly, in terms of my experience, is that it was very obvious when you were in the field with Brian that he was the happiest person in the world. And to have that as a model, and it was a great gift really to witness that for me when Brian and I worked in heronries at the very beginning. I also want to acknowledge my graduate program advisor at Rutgers. She's a formidable scientist and a very deeply committed mentor. I had the great pleasure of working in the field with her during my thesis work, as well as in some collaborative projects afterwards. And then finally, I really want to acknowledge two really special people that have been with me for good many years in my career. First of all, Dave Twitchell, just an extraordinary person, a great photographer, a Manomet trustee. He followed our little crew all the way from Delaware Bay to Chesapeake Bay up to the coast of Maine, photographing our heron work and all the crews that we had. He was a great partner in this, very cheerful all the time, and very supportive. And then my good friend Stephanie Schmidt, with whom I've worked both with Manomet and at Mass Audubon most of the years of my career. She's just been fantastic as well, very, very upbeat, and we have the pleasure of working together again now with this Boston Harbor study. Okay, let's dive into it. 3 years ago, I know you all know this, we were shocked to learn that 29% of all North American birds had disappeared since 1970. And this past spring, a global assessment confirmed that nearly 50%, 48% of the world's avifauna was known or suspected to be in decline. So this is shocking to all of us who love being in the field studying birds. But you know, the questions are obviously what caused the problem and what can we do to reverse it? Unfortunately, but you would expect this in these sort of large-scale assessments, there weren't any concrete things to grab onto and to start doing advocacy, more studies, or whatever we could do to help reverse these trends. So this is very disempowering to know that habitat loss and other factors yet to be pinned down are the cause of 3 billion birds to be lost from the continent. And if we look at some of the specific information that might have been available for long-legged wading birds, again, these large-scale assessments don't have a lot of detail, but they did estimate that the waterbirds as a big group, that about 22.5 million of those birds were gone. There have been past taxon-specific assessments of wading birds, in particular the North American Waterbird Conservation Plan had tracked multi-state sort of regional examinations and surveys of colonies from the 1970s to the 1990s. Unfortunately, there's no real coordinated recent information. But these trends and this close relationship that wading birds have with wetland resources is really what sparked a lot of my interest in the research projects I pursued. So I want to share with you today some of the long-term sublethal effects that Brian mentioned that I learned about studying wading birds in the Northeast over the past several decades. And also, just a little quick follow-up, courtesy of Mass Wildlife, we do have some more recent information just for the state of Massachusetts. And this updated information shows through 2018 that, for example, black-crowned night heron is continuing to decline. And so that only a quarter of the night herons present in the state in the 1970s are still breeding here today. So we've lost nearly 3,000 birds. And I'm going to say with hope that coordinated surveys through all these states are planned to take place in 2024. So it'll be good to get newer information. So my studies focused on these long-legged wading birds, which are, most of the species are short distance migrants. They breed colonially, which is key, usually in the hundreds to thousands of pairs at a colony site. Usually the colony sites are isolated, usually on islands. They're site faithful, generally, and they often breed in mixed species colonies with the different species using distinct foraging niches around the colony site, usually within 15 kilometers, usually no more than 20 kilometers away from the colony. And so what these birds are doing is feeding their young at the colony site from resources, prey items that they're capturing out in the surrounding landscape, and for up to 2 months after hatching. A little background on their ecology. And now in terms of sort of the frame of our studies, we conducted similar work in all of these locations along the northeast coast of the United States. We focused on breeding and foraging ecology, but we also investigated contaminants questions as they came up, which turned out to be frequently. And we developed conservation management plans in cooperation with local partners. And I want to say that, excuse me, these sites are located in lands stewarded by indigenous peoples for millennia. And of course, it goes without saying in this very busy part of our coast that every aspect of this region has been transformed since the 15th century when Europeans arrived and colonized the area themselves. In terms of field methods, we conducted colony surveys to determine abundance yearly at each colony, and at each colony we selected a 30-plus sample. Oh, thank you, Brian. Thank you. Usually a 30-plus sample of study nests for each species. One of the great things about colonies of birds is that you have access to a wonderful sample size generally, and you can have some confidence in reporting results because of that. We monitored study nests to determine hatching and fledging success. We undertook nestling growth studies. We collected egg, excuse me, blood and feathers in some years for toxicological analyses. And we assessed the use of foraging locations through surveys of those foraging habitats with radio tag birds as well, and flight line studies. So jumping in, I want to start with our studies in urban estuaries in particular. So in that 6-state region, that would include New York Harbor and Boston Harbor, but I want to focus in to begin with on New York Harbor. And the interesting thing about this location for nesting is that wading birds stopped nesting in New York Harbor in the late 1800s, and after nearly 100 years of absence, they began recolonizing the harbor in the 1970s. And this comeback followed the passage of the Clean Water Act in the United States, which essentially eliminated the hypoxic estuary conditions that were present then and allowed an aquatic food web to reestablish. So populations in New York Harbor grew through the 70s, 80s, and 90s at a rate of 15% per year. And a number of the inner harbor islands were colonized, including these two. The lawnmower is coming through the door. Maybe we're in the no, where's the numbers? Just to get back to New York Harbor, here is a very developed estuary which supported some of the most important wading bird breeding populations in the East Coast. So these islands, these 5 islands, including the 2 brother islands up in the East River, were colonized during the 1970s through the 1990s. But our in-depth studies focused primarily on the 3 western islands near Staten Island, just off Staten Island, which are down here in the west. Shooter's Island, Prall's Island, and the Isle of Meadows. And these are the common species that were the focus of our work. These are the numerically dominant species in the harbor. The annual abundance of snowy egrets, cattle egrets, glossy ibises, and black-crowned night herons generally were in the hundreds to 1,000 pairs for these 3 colonies in the harbor. And a little bit more specific ecology information. Each of these species exploits a distinct foraging niche in New York Harbor, as they do in other estuaries. 2 are fish eaters and 2 forage primarily on invertebrates. The 2 fish eaters are the snowy egret, which is really a tidal specialist in New York Harbor. It has a relatively inflexible diet of about 75% fish and then 25% Paleomenides, the little salt marsh shrimp. The black-crowned night heron, the other fish-eating species, is a generalist and feeding both in freshwater and tidal water in New York Harbor, as well as at the largest landfill on the East Coast, the Fresh Kills landfill, which I think now is closed. So it was feeding there as well. And the 2 invertebrate feeding species, one is an upland species, the cattle egret, and one is aquatic, the glossy ibis, which is also a specialist, but also is, in addition to being primarily, at least in New York Harbor, in tidal habitats, also using freshwater habitats. Okay, in 1990, very unfortunately, there was the beginning of an unprecedented volume of oil being released into New York Harbor. In all, oil in the harbor during 1990 was 150% greater than sort of the chronic release of oil for the 3 years prior. Most of the spills took place before the migratory breeding wading birds returned to the heronry, so there were wintering birds that were killed outright. But our job, because we had been in the heronries for 4 years prior to 1990, was to do a study that looked at before and after effects. So here are the 3 colonies that were our primary study sites for nesting. And here are how the oil spills kind of played out over the year. So on January 1st, 1990, a large oil spill of number 2 oil, a pipeline break very close to 2 of the colonies, then a barge spill, then another barge spill, and then tanker issues up in another waterway near the colonies. And as I mentioned before, there were black-crowned night herons and snowies and glossies and other species nesting in the harbor at this time. But what we didn't really know is that some of the most important populations from the south of New Jersey all the way to Cape Cod were in New York Harbor. Some of very significant proportions of all the birds nesting along that coastline were actually in New York Harbor. Okay, so quantifying the oil spill impacts, and this is a great field ornithology project because we had the pre-information that was pretty robust, and then we had another 4 years after the oil spills to look at a number of things, pre and post. Also, because of the diversity of the avian community, we were able to look at tidal specialists like the snowy egret and compare that to an upland species or a generalist like the black-crowned night heron. So we investigated reproductive and foraging endpoints to assess possible correlations that might occur because of the oil spills. And to start off with egg production, we found egg production. Did I kill it? Did we explode? Thank you. Such a wonderful person here. Oh, the battery died. I'll shout. Just keep moving on. It's okay? Oh, back in business. Thank you. Okay. We found, as these graphics show, that egg production for the 4 species that were our focus, the black-crowned night heron, the cattle egret, you can see their little pictures, the snowy egret and the glossy ibis, egg production, so clutch size, basically, hatching rates, and the time of hatching did not differ, or we have no evidence of seeing differences there before and after the oil spill. None of these clutch size trends vary significantly over the 8-year study. And you can see, this will happen in a couple more slides, that red line indicates when the oil spill took place. We also quantified rates and directions of foraging flights from these 3 colonies. And we found that direction was unchanged for the tidal species in particular, indicating that they had foraging site fidelity, which is something that also appears in the literature. However, we did find that flight rates were decreased post-spill for tidal foraging species, and that suggested to us that the birds were taking longer to forage in these wetlands, especially in the tidal wetlands surrounding the colonies. We also undertook shallow fauna trapping at a number of sites, 7 different wetlands following the spill. So this is information that we didn't have prior to the spill, but I think it's still interesting to look at this. So in this graph, which is one of the tidal sites, we're looking ahead possibly to a return to normal. So in 1990, this was the situation. 91, 92, 93, as conditions are hopefully getting better, we're thinking maybe that's a return to normal. But the thing that's obvious here in this graph is that the number of fish in our trapping samples was very low compared to subsequent years, and that the shrimp, the Palaemonides, primarily Crangon a little bit, were proportionally higher in that first year of the spill. And if we look at oil spill impacts to the nestling diet, these graphs compare nestling diet. We found this out through looking at regurgitations. There's a lot of regurgitating that happens with wading bird species. The parents regurgitate into the chick's mouth, and the chicks regurgitate if they get startled. Everybody is eating things up again. So we looked at regurgitations. When we put the regurgitations back in the nest with the nestlings, they invariably gobbled them up again. So that's the way they eat. In any event, we're looking at nestling diet post-spill for snowy egrets and black-crowned night herons. And you can see here, we're looking again to a sort of a return to normal. The oil spill is indicated by the red line. And if we just look at 1990, which was the really odd year, you can see that fish-eating species fed on proportionally fewer tidal fish. And the tidal species, the tidal specialists, the snowy egret, in 1990 included non-tidal organisms, which is very unusual. You can see that on the left-hand graph. And then the opportunistic black-crowned night heron in 1990 took proportionally more non-tidal prey as well as more items from the landfill. Okay, last slide on reproductive endpoints and the foraging. So for nestling production, this is a little bit of the bottom line. And nestling production, in this case, equals the mean number of young per nest that survive for 2 weeks. After 2 weeks, they start moving out into the trees and are not really available for measuring and keeping track. So nestling production was relatively stable over the study for species not specializing in tidal foraging habitats. So as you can see in the left-hand graph, we're showing information for both black-crowned night herons and cattle egrets. And black-crowned night herons are producing about 2 chicks per year per nest on average through that entire period. Cattle egrets there's some variability, but cattle egrets are producing about 1 chick per year through that period. And if we shift over to the right-hand graph and look at the information for snow egrets and glossy ibis, you see that the oil impacts seem to be pretty severe, and they lasted for 3 breeding seasons, because it's clear after the oil spills that their production of nestlings went way down and persisted, unfortunately. So not just a one-year effect. So what we found is that nestlings were lost primarily as a result of starvation. We were weighing and measuring young twice weekly and they were marked, and this helped us to determine the primary factors of mortality for nestlings. And you can see that prior to the oil spill, the proportion of young that we knew did not fledge were lost kind of 50-50 to starvation and predation. But for snowy egrets, starvation accounted for significantly more deaths in 1990 than in pre-spill nesting attempts. And you can see that that proportion of starvation declined over the following years, indicating, again, something like a return to normal. All right, a shot. Okay, so to summarize, in terms of productivity, we found that egg production was unchanged. We found that tidal specialists produced fewer fledglings 3 years post-spill, so for a time, a few years. In terms of mortality factors, nestlings of tidal specialists grew slowly and more starved. And for foraging, we found that the diet of the generalist species, black-crowned night heron, included fewer tidal prey. The tidal specialist included fewer fish and more shrimp, and foraging time increased. And then finally, with habitat, foraging habitat, there was a decreased abundance of forage fish in tidal creeks post-spill, and relatively more shrimp, which are actually hardier in oil spill situations. Okay, so before we completely leave this urban setting, I wanted to talk briefly about another class of contaminants that we more typically associate with legacy situations in industrialized estuaries. And I would say again that oil contamination is typically a chronic source of habitat degradation in New York Harbor and elsewhere. However, in 1990, as we saw, that there was an extraordinary dosing of wetland habitats, which caused fairly extreme reductions that lasted for 3 years in heron productivity. But this other class of contaminants that is part of the chemical background, especially in industrialized estuaries, is the presence of organochlorines. And organochlorines in New York and Boston are manufactured locally. There's a big industrial complex there in both of them, especially New York. So organochlorines, as I'm sure you're all aware, are embryotoxic, and they're long-lasting chemicals. So they're part of, as I say, the chemical background. And if we look at these data here, you can see that I'm showing you information about DDE, which is a metabolite of DDT, PCBs, which are industrial chemicals, and then 3 versions of chloridane, which is an organochlorinated pesticide, all produced in New York Harbor. So these, and other places. So these data show for 5 different locations. We have Nantucket here in Massachusetts, Delaware Bay, far south, Boston Harbor, Cape Cod, and New York Harbor. The surprising situation here is that Cape Cod comes up high. It's sort of it comes in second place, unfortunately, and that's sort of surprising. But the other 2 non-urban estuaries, Nantucket and Delaware Bay, are very significantly lower than the levels of these contaminants that we found in Boston and New York Harbor. And I would also say, just in closing on this slide, that there are 2 Superfund sites on Cape Cod in the Upper Cape area that are associated with the historic use of the region for a military base. Okay, and then sort of simplified charts to show how hatching success played out in an urban site compared to a non-urban site. So you can see here from these simple pie charts that inviable eggs accounted for about 3/4 of all the eggs that were known not to hatch. So we know that organochlorines are still present in the eggs and in the environment. They have long half-lives, 60-year half-life for DDT, for instance. And they're embryotoxic, as I said, and you can see that egg inviability is really an urban problem here. In these pie charts, just to back up a little bit, you can see that I'm comparing for snow egrets, the relative proportions of eggs that were predated versus ones that didn't hatch and lasted past their hatching date from Boston, comparing Boston to Delaware Bay, both for snowies and black-crowned night herons. Okay, so now I'd like to shift, because we're starting to look at comparisons across the entire 6-state region to suburban and agricultural estuaries. And these included a large colony in Delaware Bay with extensive agriculture in the nearby landscape. The nearby landscape within 15 kilometers of the colony was about 35% in crop agriculture. As well as some of the other non-urban sites that we studied included, as I mentioned earlier, Cape Cod and Nantucket, which are really dominated by residential development, as well as cranberry growing. So jumping right into problems that we detected early on in the study, we began seeing lesions on the undersides of black-crowned night heron and snowy egret chicks in this colony on Cape Cod. This was first observed in 1990, and then when the study expanded to Delaware Bay in 1993, we documented lesions on all heron species in all estuaries. So even in the urban estuaries, we were seeing lesions. And you can see from this slide that they could be a pinprick kind of a lesion, like the chick in the upper left of this slide. The center picture shows lesions healing, and then the other two pictures show actively bleeding lesions for these chicks. And if you look at the percent of nests that had chicks with lesions, you can see that these are fairly significant percentages for black-crowned night heron, 88% in the Cape Cod colony, Delaware Bay, 30%, and then for cattle egrets, which didn't occur as far north as Massachusetts, 45% of nests had birds with lesions. So, because there was such variability in the size and the severity of the lesions, we developed a lesion score to try to accommodate that variability. And when you look at the lesion score for all of the nests that or all of the birds that we examined, you can see here in this table that for black-crowned night heron, snowy egret, and you can all those abbreviations are for black-crowned night heron, snowy egret, little blue heron, cattle egret, and glossy ibis in the 4 major regions where we were studying them. You can see that, again, the importance of lesions on black-crowns in Cape Cod, and then the importance of lesions on cattle egrets in Delaware Bay is really where we're seeing a big problem. However, as I mentioned before, we found lesions on chicks in all places. It's just that in New York Harbor and Boston Harbor, they were mostly those little pinprick lesions, and for the other species, they were of smaller concern too. So they were rarely seen in New York Harbor or Boston Harbor. They were rarely seen on little blue herons, and they were never, not in one, not thousands of nesting examinations, were they seen on glossy ibises. All right, so we began questioning this, and started some investigations into what was causing lesions, and we looked at nest construction and presence of irritating materials that might be in the nest, briars or thorns or what have you. So we collected, under appropriate permits, after the end of nesting, about 250 heron nests from 5 species, 7 colony sites over about an 8-year period. So there wasn't a massive removal of nests in any one year, from Delaware Bay to Boston Harbor. We deconstructed all of those nests. We identified the twigs to species if we could. We counted all of the ones with briars or thorns, and tried to answer that question: was there irritating material in the nest that could have been causing these lesions? But we found no evidence that that was the case. What we did find in these collected nests were dermestid beetle larvae. And dermestids are scavenging beetles, and what we found in the nests both during active nesting, sometimes we would see them right in the nest, were caterpillars, were caterpillars of this species. And Dermestes nidum was found in 90% of the nests from all locations, both with lesioned birds and unlesioned birds. So they were part of the background. They were part of what the bird, part of the whole ecology of a heron colony. We found ultimately that the number of beetles in a nest correlated most strongly with local annual precipitation, but not with any of the other sort of obvious variables that we tested, such as herring species, who was using the nest. They use old nests, they kind of switch things around. But anyway, there's a lot of moving around in the heronry, both between years and within the year, depending on nesting success. But also colony size, location, all of those things didn't help understand why we were seeing variability up to 300 beetles in a single nest. The number of beetles average around 30 per nest, but again, we did take apart a nest that had 300 of these caterpillars. And then somewhat surprisingly, we found that there is no evidence that beetle abundance was related to lesion occurrence. So that was a head scratcher. So at this point, we believe that nest material did not explain occurrence of lesions. The possibility of disease or immune status was related to lesion occurrence wasn't supported when we looked at white blood cell counts of sampled lesioned nestlings. In addition, in Delaware Bay, we were able to substitute insect-free artificial nests for cattle egrets that had active nesting. They had nestlings in the nest. So we took the nest, put the little pie plate in there, a little straw pie plate, and that had been put in an oven for a period of time. So we knew there were no beetles in those artificial nests. Put those in, monitored those nests and all those nestlings, never recorded lesions on those nestlings. So we also documented that when nestlings began spending more time out of the nest, branching, which is what they do when they don't want to wait for the adult to come back and feed them in the nest, they want to be up at the top of the tree and intercept the adults before they get too far. When the birds are branching, we found that those lesions usually healed, and in fact, lesions were rarely associated with direct mortality. But what we did find, and this was because of a collaboration with the Tufts Wildlife Clinic and the legendary generosity of Mark Pokris. I hope everybody, or I hope all of you have a chance to interact with Mark. In any event, his vet students assayed blood samples for cholinesterase and found that lesion chicks had comparatively low levels of cholinesterase compared to unlesioned chicks. Cholinesterase, this is something I learned because it's not a field ornithology topic necessarily. Cholinesterase is a fundamental molecule that's common to nearly all animal species, including insects, birds, mammals. So we all have cholinesterase, and we need cholinesterase to have normal neurological function. It's an enzyme that catalyzes nerve signal transmission, and if you don't have enough cholinesterase, your neurological functioning becomes impacted. And so we found this significant relationship with lesioned and unlesioned black-crowned night herons and cattle egrets, where cholinesterase was low in lesioned birds and higher in unlesioned birds. And furthermore, looking at a larger scale, we found that mean cholinesterase levels in non-urban estuaries were lower than urban estuaries. So comparing those two groups of chicks. Okay, so then looking in the toxicological and biochemical literature, we tried to understand what the possible causes of depressed cholinesterase might be. And disease, nutritional deficits, mercury contamination, and a whole class of pesticides that are known as anti-cholinesterase insecticides. So we assessed immune status, again, through our white blood cell counts, nestling growth with our growth studies. We worked with a number of labs, environmental labs looking at mercury in blood. None of those things provided evidence that they were impacting cholinesterase. However, we did find through residue analysis that insecticides were detected in prey items as well as in leg washes of cattle, of adult cattle egrets that are feeding in farm fields. And we also, through Mike Hooper's lab, performed reactivation analysis, which is essentially a test where your cholinesterase that's lowered because of an organophosphate or a carbamate insecticide can be reactivated by basically applying an antidote that pulls the pesticide off the cholinesterase molecule, and when you reassess those levels, the cholinesterase is higher. So that's a reactivation analysis, and specific for organophosphates or carbamate pesticides. So if you get reactivation of cholinesterase after adding the antidote, you know it was an organophosphate that was keeping the cholinesterase down. Okay. So partnering with environmental chemists at Southern Illinois University, these are the pesticides that were found in prey items and from the leg washes of cattle egrets. And again, for context, these are second generation pesticides that are used on crops and mosquito control, termites, pet and lawn care. They became the go-to pesticides in the early 1970s when DDT and other chlorinated pesticides were banned from use in the United States. So 20 years ago, 1.2 billion pounds of anticholinesterase pesticides were applied in the United States, and that's more than 20% of the world use. And also, to show the importance of this, going back. Well, showing the importance of this is really that in 20 years ago, 75% of all the pesticides that were used in agriculture were in this anticholinesterase class. And then the last thing that I wanted to mention is that 20 years ago, again, it was documented that 80% of homeowners were using pesticides of some kind, you know, in lawn care and taking care of their pets, termites and all of those things. So there's a lot of chemicals out there, and I know everybody's aware of that. It's better now, actually. In the 1990s, two of the most harmful organophosphate pesticides were also very severely restricted in the market, chlorpyrifos and parathion, so that's a step in the right direction. Okay, we're getting close to the end of the pesticide story. What we wanted to do was develop reference values for cholinesterase. We know that cholinesterase was different in lesioned and unlesioned chicks, but we wanted to know what's a reference value? What would chicks where we're controlling the diet and controlling all of the factors, what would their cholinesterase levels be? And so we teamed up with Tri-State Rescue and Research, a great organization in Delaware. We brought in hatchlings of cattle egrets and raised them on a pesticide-free diet and determined this reference value, this mean reference value. And you can see from this graph that the reference values are higher than the wild birds without lesions, which is in turn higher than the chicks with lesions, the wild birds with lesions, excuse me. So that was interesting. Not only were the lesion chicks had depressed cholinesterase, but the whole population in the wild was depressed compared to the reference values. Okay, this is the summary. So what's the evidence for insecticide exposure? Residue analysis, reactivation analysis. Cholinesterase of wild nestlings is lower than captive broods. Birds in farm habitats are showing the greatest effects, cattle egret. Greater effects in agricultural estuaries compared to urban estuaries. And also, you know, fairly diligent quantification of other possible factors provided no evidence that they would explain what we were seeing. So we had determined that cholinesterase was depressed in these wild birds by exposure to pesticides. And we believe that nestlings were incidentally chewed on by beetle larvae. But they rarely succumbed to these beetle-inflicted wounds. And we wanted to know if there were other impacts to the chicks with lower cholinesterase, because it's such an essential part of being a wild bird. You know, your neurological system's got to work pretty well. So from the avian toxicological literature, again, we found out that low cholinesterase in laboratory studies and what have you was associated with lethargy, with decreased nest attentiveness, with loss of predator avoidance behaviors in general. So this played out, as you might have expected, in an increased risk of predation due to depressed cholinesterase, we believe, and it showed up dramatically in how nestlings were lost. So these are nestlings, again, the simple pie chart, that help us understand the factors of mortality that are impacting nestling success. And the primary factors are starvation and predation of nestlings. And you can see, again, this comparison between urban black-crowned night herons and urban cattle egrets to Delaware Bay black-crowned night herons and cattle egrets. And you can see that predation and starvation are accounting for approximately equal proportions of all the nestlings that were known not to fledge, both for cattle egrets and for black-crowned night herons. But when we move into the non-urban estuary in Delaware Bay, we see that predation is accounting for at least 3/4 of all the lost nestlings. Oh, and also I should mention that in these colonies every year, we also documented how many predator species, avian predator species, were nesting in the heronry, with the wading birds. And so that included owls and crows and common grackles. And we found no evidence that density of predators that were nesting along with the wading birds, impacted these predation rates or contributed to this lopsided predation rate. So that we're convinced that this scenario explains what we documented over many years and in many locations. We think there's chronic exposure from low-level dosing that is resulting in these sublethal effects, both the increased predation rate as well as lesions. And it's important to know, because I think this is how you can have 3 billion birds lost over a 2 or 3 decade period and not know what happened exactly. This low-level dosing and also with the oil spill and the chronic exposure to organochlorines is not resulting in piles of dead birds that anybody would necessarily come across, but really impacting predator avoidance behaviors or just reducing hatching and causing these losses in reproductive success. Okay, so how do we swing that back around to matters of heron abundance and distribution? Excuse me. So looking between the 1970s and 1990s at large non-urban colonies, we have 2 graphs here, Pea Patch Island, which was at the time the largest heronry east of the Mississippi River, lost about 15,000 pairs of birds, from its heyday down to the early 2000s. And then Stone Harbor, New Jersey. I don't know if any of you had been there, maybe not. A very wonderful, accessible to the public heronry along the coast of New Jersey. That was New Jersey's largest heronry, and 5,000 pairs of wading birds were lost from that colony over a period of decades, the 70s to the mid-90s, it looks like, mid-80s. At the same time, as I mentioned before, urban colonies were increasing. So New York Harbor was increasing 15% per year from the mid-90s, or from the mid-80s to the mid-90s, and Boston Harbor as well increased by more than 400% from the 70s to the 90s. So these were really fantastic upswings in abundance. And I just want to reiterate the importance of these metropolitan populations to the entire region's populations of wading birds. By the 1990s, New York Harbor wading bird populations were significant for the entire 3-state region of New Jersey, New York, and Connecticut. So for example, for black-crowned night herons, almost 50% of all the night herons in that area, in that 3-state region, were nesting in New York Harbor. So very vulnerable in that respect. And similarly, in Boston Harbor, nesting grew from 8%, this is the little blue line in those 3 bars, so went from 8% of the entire state population in Boston Harbor to about 50% by the 1990s. So the metropolitan populations became real mainstays for all of the herons that were nesting throughout this region. And one final interesting piece of information, there were about 50 heronries in the 6-state region, Delaware to Maine, in the 1970s, and also in the 1990s, about 50 heronries. And you can see from these simple pie charts that in the 1970s, about half of those 50 heronries were on true islands that had a water barrier around, and about half were on relatively isolated sites, like the photograph below that is Coatue on Nantucket. So it's a very long way from, from cats and sort of human development to get to Coatue, but there is a land bridge to that heronry. And so about 25 of the heronries in the whole region had some kind of land bridge to the colony, whether it was at low tide with tidal habitats or what have you. And so they were not as isolated. But by the 1990s, almost 90% of those 50 heronries are now nesting on true islands and have that water barrier, which is keeping ground predators from accessing the sites for the most part. Okay, looking kind of at the bottom line at sustainable or unsustainable rates of reproduction from the 1980s through the 1990s. We had collected extensive data, as you had been, I've told you, over this 10-year period for nesting sites from Delaware Bay to Boston Harbor. We documented that fledgling production was not sufficient in almost all cases to sustain populations at these locations. And the sustainable rates, which I'll just point out here, are the very dark blue bars in this histogram, and all the other ones are sites that we measured reproductive success. So clearly the orange, which is Delaware Bay, is very low compared to the sustainable rates for snowy egrets, black-crowned night herons, and cattle egrets. But just in general, Delaware Bay is very low. The other two are intermediate until you get to Boston Harbor, and only for black-crowned night herons in Boston Harbor is the level of reproductive success matching and kind of like slightly exceeding what the sustainable rate is thought to be. And then moving into the decades past the 1990s, and again, the 1990s, the information contained in the North American Waterbird Conservation Plan goes through the 1990s, it was published in 2002. You can see, unfortunately, that for New York Harbor, we have site-specific information that those populations declined after peaking in the early 1990s. And similarly, the proportion of Massachusetts wading birds that were nesting in Boston Harbor also declined up until 2018, which again, we have some preliminary data courtesy of Mass Wildlife to show that that trend there. So herons are disappearing from those two urban estuaries. And then finally, also not very encouraging, from our 2-year study we're finishing up this year, there's very preliminary data from Boston Harbor. And you can see that fledgling production, again, the number of fledglings or nestlings that made it to 15 days post-hatch, is only 1.1 in 2021 for black-crowned night herons in Boston Harbor, whereas in the 1990s we published that 2.8 on average chicks were making it to 2 weeks post-hatch. So that's not encouraging at all. You can see that predation, kind of unusually, is accounting for a lot of the lost eggs and lost young. And one thing that we noticed in these more recent heron studies is that the predator, the predator context is really quite different. There are coyotes that are swimming to some of these islands. We have urban coyotes. They're more of a presence. Great horned owls are nesting right in this particular colony. Bald eagles are hanging out in one of the colonies in the outer Boston Harbor region. So there's a very big stress, I think, with predators, and in some cases, eggs and young are being predated before we could find out if maybe they would be inviable, or if there could be other factors that could be impacting them. So final couple slides. The declines of these small herons in the Northeast greatly surpass the average loss of those disappeared birds in the 2019 science paper by Ken Rosenberg and his colleagues. So 62% of black-crowned night herons lost from the 6-state region, 63% of snowy egrets, 92% of cattle egrets, and cattle egrets are more marginal in terms of their geographic distribution, but very significant declines for those three small herons. However, it's very hard to summon optimism for me for this record of loss, especially because I'm retired, and so it's hard. I'm hoping all of you will step up and try to recover some of these populations, and I'm definitely there supporting you. So it's hard to summon optimism personally, but there are definitely some bright spots, and I think there's reason for hope. Certainly there are many questions about the species that were apparently stable. And again, the data are old, but they were apparently stable or increasing in the 1990s. The glossy ibis, for instance, is a terrestrial ornithid. It's not an ardeid. All of the heron species were showing impacts from low cholinesterase. But the glossy ibis didn't. We never saw lesions on them. Their cholinesterase, it was quite high, and it turns out that as an ibis, they actually have higher background levels or baseline levels of cholinesterase, and therefore might be more buffered against the anti-cholinesterase pesticides. Also in the region that's reported here, and again, this is the 1970s to the 1990s, it's the Manomet region of the North American Waterbird Conservation Plan, so it extends into Virginia and into Maine, and so the timing and the geography is a little different. So little blue herons are comparatively small population, and they might be subject to the kinds of exciting plus 53% numbers that you might see with a small population that's really concentrated in the very southern parts of this larger range. So I'm not sure about that. Great blue herons, as you probably know, have benefited massively from limited trapping of beavers in the last several decades. So they're going great, and throughout, more freshwater habitats, of course, but there's lots of great blue herons. And then I would kind of end, let's see if I can do this. Nope, that's not what I want to do. Okay, I would end by saying that the great egret is a question mark, right? I hope you've all been seeing more and more great egrets in the wetlands here or wherever, maybe wherever you're from. They're fairly widely distributed. I don't know why they're increasing and have been. I have some ideas, but it's a little bit of a mystery. And I would say that it's emblematic, or they're a great sort of species for hope, because they were a very specific target of plume hunters at the end of the 19th century. And their decimation in those decades led to the enactment of the Migratory Bird Treaty Act a little bit more than 100 years ago. So with that, I'll leave those ideas with you at the 100th anniversary of your association, and let you sort of ponder some of those interesting connections. But before I finally end, I want to acknowledge the many field ornithologists at the beginning of their careers who stepped bravely into the poison ivy and climbed into many trees, and contributed, very importantly, their insights and ideas to this work that we did very collaboratively. And also the site partners who entrusted us with diagnosing problems in their colonies and developing management plans for their future success. And then, of course, many agency and foundation funders made the work possible. And then finally, I want to acknowledge and thank my family. They participated in the work. I had little ones that were helping out in the field. They participated in the work, and they greatly supported me, so I'm very, very grateful to them. And with that, I think I've taken quite a bit of time, but if there's time before the coffee break, and I'll be around if people have questions. If there's time for questions now, happy to answer any. Do we have time for questions? Yeah. Oh, okay, great. Scott.
Wonderful presentation, Katharine.
Oh, thank you.
Feel free to speculate about great egrets.
Well, You know, it's interesting. They forage in deeper water. They're larger, and therefore forage usually in deeper water. They're only 2 to 5% of all the great egrets in the world are in our region of North America. So there are a lot more in other places, and there might be some of that starting small and making big changes at the beginning of the trajectory. But I don't know. What do you think?
I have no idea.
What does everybody think? Brian, what do you think?
I never think.
Always question. It's a good question. I don't know, but it's a great sign to see those beautiful birds, and I see it's on your newly designed journal cover. So it's a great symbol of hope over all these decades and decades that the great egret is increasing in North America. Yes.
Could you comment on your experience sharing this long-term data set with decision makers? Because it's so impressive and comprehensive.
Can you repeat the question?
Sure. Oh, yeah. Great. Thank you so much for that question. That gives me a chance to laud Manomet and its unusual kind of way of operating. So the question was, can I expand a little bit on how this information hopefully impacted decision makers and how pesticides or oil spills were handled in the future. And I would say that for the oil spills in particular, the Oil Pollution Act was enacted just at the time of these major spills in 1990. And the benefits of that act, which changed how many years pilots had to be trained before they could bring oil barges and oil ships into narrow harbors like New York Harbor, as well as double hulling and things like that. Those big changes following on the Exxon Valdez spill, I think have made a big difference in oil spill prevention. In terms of the pesticide problem, which is kind of clearly a problem not just of agriculture, but also of lawn care and small-scale crops. Manomet had this wonderful way of having us work with stakeholders. So we wanted science to be in the middle of a conference table that farmers and mosquito control operators and citizens and wildlife managers would all be around looking at the information and deciding what to do with it. And so we actually did that in Delaware Bay. We sent our raw data so that they could calculate their own coal esterase values and look at the data themselves to the mosquito control agencies. And in Delaware Bay, we were already working in a framework that included agencies in New Jersey and Delaware, and they were very supportive of having this information help manage not just the colony, but also the surrounding landscape and the impacts to wetlands. That was actually going well. The mosquito control management had changed because of this information and had reduced spraying in some ways and certain chemicals. Unfortunately, West Nile virus then raised its head, and human health concerns, rightly so, you could argue, superseded any of the changes that we had accomplished with mosquito control. So, yeah. Any other questions? Yes?
So I was wondering if you could comment on this increase in predation, because I was wondering if you think it's more of a sign that maybe the environment's getting, the ecosystem's getting healthier, or is it like they are getting pushed to these areas from the out of location?
So the question was, is there more elaboration on the increased predation that we're seeing right now in Boston Harbor? And, yeah, okay. And I would say that it's the latter of what you suggested, which was that there are new predators in town, basically, and there's a new mix. And I think it has to do with the fact that and we've certainly found this with piping plovers and ground nesting birds in our developed coastline, that there are more coyotes, of course, around, and there are more raccoons, and there are more great horned owls, and now there are more bald eagles, and so all kind of wonderful. Coyotes have moved into the urban areas. They're right on the Boston streets and many other streets, city streets throughout the continent. So I think that partly explains what's going on there. The colony that we were studying intensively in Boston Harbor is a very small island, and it's kind of the only show in town for nesting herons, unfortunately, because the other islands that were supporting them, they've all disappeared basically. So it's a very dense nesting area and very subject to a predator, a coyote swimming onto the island. We've put out a lot of cameras, and they're deer, they're coyotes, they're rats. There's just a lot of predators out there. Any other questions? Yes.
I have a follow-up question. So there's this increase in predators. Do you notice even anecdotally if the species have different predator behaviors that may benefit or imperil the nest? That could explain some of the differences.
So, very interesting question. So the question is, are we noticing new or changed anti-predator defenses that in our most recent study in Boston Harbor? And I can't say that we have. At least not anecdotally, we have not analyzed all of our camera data or even looking at nest height, which I think is key for predator impacts. And because the colony is so densely packed, and in the canopy are double-crested cormorants nesting and causing the vegetation to really degrade, I think that habitat availability and the availability of nesting substrate is definitely part of the mix. Thank you so much for your attention. I very much appreciate you being here this morning.
Just one thing. If you like these mugs, there are a few of these at the Avian desk for purchase. So you can go there and purchase some of these orange mugs too. Thank you.